Fantastic. So yes, as Lars mentioned, I work in his lab as a PhD student in microbiology. So this will be a little change up from the last few talks. I'll be presenting some of my most recent work on characterizing this interaction between Rhodopseudomonas palustris and Geobacter inside of a microbial electrochemical system for the conversion of lignin to electric current. So for anybody who's read any of Lars's papers for a long time out, our lab has always been very interested in anaerobic processes. So these can be very widely distributed, and they have many advantages, usually conferred by an environment where the availability of terminal electron acceptors is very low. And pictured here could be an example of one of those processes such as anaerobic digestion, where you can feed in diverse waste streams and get a harnessable product out from this bioprocess. In this case, it would be methane. However, one application of anaerobic processes that doesn't work very well is the degradation of lignin. And it was actually believed that degradation of lignin did not happen at all under anaerobic processes up until this was demonstrated in a methanogenic enrichment culture back in the 1930s. Even then, this process doesn't happen efficiently, doesn't happen very rapidly. And it was something that we wanted to look into a little bit further. So right here, I had it. Oh, there we go. There's some reactor digestate coming out of an anaerobic reactor. And lignin is actually one of the primary components in what you'll get left over from anaerobic digestion. So it just attests further to the fact that this very complex, undefined, recalcitrant molecule is really tough to degrade under anaerobic conditions. And here, this is just a generalized structure of it, but you can see that it's usually repeating monomers of some phenolic derivatives. So with that in hand, what we wanted to do was work on a biosystem for the degradation and potential conversion of lignin into a harnessable product. And since I'm here, this relates to microbial electrochemistry. So for a basis, we wanted to start with an organism that could be suitable for this process. And one of the prime candidates for this is Rhodopseudomonas palustris. It's a model organism, and it's primarily a model organism for its incredibly diverse metabolism. It's actually currently characterized as the most metabolically versatile organism that we know of yet, right? So, and one of its model features is the degradation of lignin, a wide range of lignin monomers under anaerobic conditions. Key to this, it's only capable of degrading these monomers under fully anaerobic conditions and in the presence of light. So what we have here is a very common and one of the substrates that Rhodopseudomonas is characterized for, paracumaric acid. And this is one of the largest components of actual complex lignin. Now obviously this is not lignin itself, as I showed on the previous slide. So for this study it'll be a simplification of lignin degradation. And this compound is rapidly metabolized by Rhodopseudomonas to this intermediate here by easily cleaving this 2-carbon group from the side chain, and then it's further degraded into this kind of common metabolite benzoate. And from benzoate, the ring is activated, cleaved, and then it's kind of degraded in a beta oxidation manner. So with that in mind, as a microbiologist, I feel like I have to give a little bit of a crash course in some of the notable features of Rhodopseudomonas' diverse metabolism. The first, as I alluded to before, this has been a reoccurring theme, yeah? Where's the receptor? There we go. Oh, great. Okay, so anaerobic anoxygenic photosynthesis is used to provide energy for any processes that it wants to do under these conditions. In addition to that, it's got this great versatile metabolism where it can take up these lignin monomers and integrates them within its central metabolism in the form of acetate. As recently described, it is also capable of photoferrooxidation, which is capable of coupling it to a biocathode, as was described by the Gerges lab recently. In addition, it's capable of nitrogen fixation under anaerobic conditions. Great. In the absence of nitrogen, however, it's capable of using its nitrogenase for the evolution of hydrogen. I'll come back to this issue. And in addition, it's capable of carbon fixation as well. Now, besides the typical view of nitrogen fixation and carbon dioxide fixation in growth, these two metabolic strategies are actually quite important for Rhodopseudomonas in novel ways. And those are in the sense of maintaining redox balance under these conditions where terminal electron acceptors can be depleted. So, to highlight that, here's Rhodopseudomonas growing under these anaerobic conditions with an organic substrate. And when it does this, the reducing equivalents that it gains from these substrates are in excess of what it can utilize for its central metabolism. And because of that, it finds novel ways to get rid of reducing equivalents to maintain redox balance. So, for example here, CO2 is being fixed into biomass not only for growth advantage, but actually it's functioning as an electron sink. And that's illustrated here as well, where you could complementary substitute these reducing equivalents into the nitrogenase and evolve hydrogen as well. So what this is really suggesting is that these two strategies play an important role for central redox balance in this organism. To further highlight that is a graph here shown. On the left-hand side is a wild-type strain of Rhodopseudomonas. On the right-hand side is a mutated strain that constitutively expresses its nitrogenase. Below the x-axis here is the influx of reducing equivalents in its metabolism, and above is where it deposits those reducing equivalents. So we can see here that when Rhodopseudomonas is constitutively expressing its nitrogenase, a large fraction of the reducing equivalents are actually being sunk into hydrogen, as opposed to being refixed into carbon dioxide for the wild type. So with that in hand, the big picture is essentially we have this organism under anaerobic conditions, gaining energy from light, taking up a potentially recalcitrant molecule, converting it to central metabolism, and producing hydrogen at the same time as a strategy for maintaining redox balance. And based on the title of this talk, you can probably guess what this is coming to. If we have hydrogen production, we have some leaky amounts of acetate escaping from this metabolism. This presents a great opportunity to couple these two substrates into Geobacter and integrate this redox balance into a defined co-culture where the reducing equivalents actually end up at an electrode through the high-efficiency metabolism of Geobacter. So with this large picture in mind, these were the experimental conditions. It was done in a H-type 3-electrode system with a poised working electrode via potentiostat, defined conditions, 30 degrees, incandescent illumination, and passive anaerobic working chamber, and a sparged counter chamber. And the concept was to pre-grow a Geobacter culture and get it sustained and viable on the working electrode, replace all of the growth media from the Geobacter culture with a FW paracumarate culture, paracumarate media composition, and then inoculate with our Rhodopseudomonas nitrogenase expressing strain that we know makes hydrogen under these conditions to see if we could recover some of these reducing equivalents from this lignin monomer. And so essentially what we have here is Geobacter was pre-grown in this region. The culture was washed through with 5 volumes of the cumaric acid media, and we established a baseline and inoculated Rhodopseudomonas into the media. These are 2 biological duplicate reactors. Below here is a control reactor with just Rhodopseudomonas in these conditions, no Geobacter co-culture. So basic reactor characterization we see here upon inoculation. There's a slow induced growth over time of the planktonic phase, and this is largely attributed just to Rhodopseudomonas since Geobacter is remaining isolated to the biofilm, and small deviations in pH. If we're looking at the metabolite breakdown, as we would expect, the first thing to be degraded is the percumaric acid. This side chain group is cleaved off, and we see that happening almost exclusively first. That goes along with the onset of Rhodopseudomonas Rhodopseudomonas OD and the very beginning of current production from Geobacter. we see the emergence of this intermediate species, 4-hydroxybenzoate. The aromatic moiety is much more difficult to degrade. It requires activation before this complex can be degraded, so this side chain is preferentially degraded first. But what we see here, upon the transition of Rhodopseudomonas' metabolism from this substrate to this substrate, there's kind of an inversion in the current produced by Geobacter, demonstrating that there's a large interaction between this metabolism form in Rhodopseudomonas and what Geobacter is getting at the electrode. So this was a single batch experiment. If you go on to repeat multiple batches in this case, flow through new media, and just allow the Rhodopseudomonas that remains inside from the initial batch to continue to grow, we'll see that we kind of get a diminishing return on our invested carbon in this case. And there's rapid consumption of the initial compound here. And basically, what this is coming down to is, as we get more and more Rhodopseudomonas residing inside our reactor, forming into biofilms on the electrode, forming into biofilms on the reactor surface, there's a lot more cells that can be in there metabolizing, and they convert the substrate over more rapidly. To make sure that somehow in the process of this co-culture, Geobacter wasn't inhibited by some product, by out-competition by Rhodopseudomonas, at the end of this triplicate batch experiment, just acetate was spiked into the reactor to verify that Geobacter still had a functioning metabolism in this case. We can see a large immediate response from Geobacter, signifying that within this co-culture it's still healthy, it's still capable of scavenging any metabolites that would come its way. So this signifies really that the limitation in current here is because Geobacter just isn't seeing these reducing equivalents that are being released from Rhodopseudomonas' metabolism. Going back, if we're interested in calculating Coulombic efficiency for this process, as we would expect, we see diminishing returns with each sequential batch. Starting somewhere around, around 6% for the initial culture, with subsequently less each time. And so this really brings us to a question of why are we getting these diminishing returns with each continuous batch? It's hypothesized because Rhodopseudomonas has such an incredible diversity of its metabolism, it's capable of doing many different things in one environment. And so there's a hypothesis out there that within these lignin monomer degrading pure cultures, Rhodopseudomonas actually forms sort of a semi-syntrophic relationship with itself, where one subpopulation is responsible for the primary degradation of the molecule, and another subpopulation is actually complementing it by performing these redox sinking activities such as the CO2 fixation or hydrogen evolution or something like that. So they're partitioning tasks between the same organism within this culture. So our idea, in essence, was essentially, okay, instead of replacing this syntrophic partner of Rhodopseudomonas with itself, if we put Geobacter in there, maybe that will function just as well. And we see that it does function within the first batch. However, when Rhodopseudomonas starts to become more prevalent inside the system, it becomes uncoupled, potentially, from this interaction with Geobacter, where it's taking care of it's excess reducing equivalents. And Rhodopseudomonas starts to function again with itself, potentially as its own syntroph. And with that, that would somewhat explain, as we do the sequential batches, as more biofilm of Rhodopseudomonas emerges, we're getting diminishing productivity from Geobacter. So to highlight a few of my findings, we found that Rhodopseudomonas is capable of providing a good substrate for Geobacter when growing on this lignin monomer. And the current produced by Geobacter coincides with the major metabolic stages that Rhodopseudomonas goes through in these processes. Geobacter wasn't inhibited within this co-culture. But as you can see here, Rhodopseudomonas is really great at growing under these conditions, and it forms these really, really thick biofilms on any surface that's exposed to light. So over time, the Coulombic efficiency provided by Geobacter is somewhat outcompeted by the presence of more and more Rhodopseudomonas. So with that hypothesis in hand, there's a few things that in theory could be done to improve the performance of this system. One could be by potentially operating in an environment where CO2 is not a potential electron sink for Rhodopseudomonas. In this case, they're forced to divert their reducing equivalents to an alternate route, maybe minimizes the importance of the potential pseudosyntrophic relationship with itself. Because this process is also tied to illumination, potentially modulating light in some sort of regiment or area could be utilized to improve Geobacter's performance. And obviously, improving reactor geometry so that the electrode to illuminated surface area volume was more beneficial for the Geobacter side of this reaction. So with that in mind, I'd like to make a few acknowledgments to the Angenate lab at large, of course Lars primarily, but to everyone who was part of the microbial electrochemical team at the Angenate lab over the past few years who have all contributed a lot to our progress. And with that, I'd like to field any questions.
3 minutes of questions.
Yeah.
Have you thought about re-inoculate the Geobacter again after you got the co-culture, that one to fade away so that Geobacter performance will not dispense electron transfer?
Sure. So actually, I've done this. Re-inoculating Geobacter doesn't do very much. The Geobacter biofilm is still on the electrode. It's still intact. It's still functional, but it's just substrate limited. And that's also demonstrated when you add in excess acetate. Geobacter is able to rapidly uptake that and perform its normal metabolism. So it's not so much, it's been outcompeted on the electrode surface as far as area, but it just doesn't have the substrate. Yeah.
Okay, thank you. So from your data, it seems like all the current are coming from hydrobenzoate. Because they metabolize coumarate and convert it to hydrobenzoate, and you generate the current.
So current induction starts with the conversion of the paracumarate. Right? That's when we see, upon the change from cumarate to 4-hydroxybenzoate is when we see that inversion in current. Now, at the very beginning, the Rhodopseudomonas biodensity is very low because it was just inoculated in. So the actual flux of consumption is lower as well through cumarate than it is later on in the batch through 4-hydroxybenzoate. So that's why the current is highest at 4-hydroxybenzoate, because that's finally when Rhodopseudomonas has reached its highest concentration. Yeah. Yeah.
Have you considered, apart from the strategies that you suggested to control the proliferation of Rhodopseudomonas, a third microbe that could be a gatekeeper on each end that would probably kind of convert the products from one to the other? Since in anaerobic systems, we know that they might have benefits with multiple cultures.
Sure, sure. I haven't thought too much about that, no. But that would be something to consider for sure.